Welcome to BioBits and Bytes. Today, we're diving deep into the world of microbiome bioinformatics. Think of it as using powerful digital tools to understand those incredibly complex communities of tiny organisms, microbes that live, well, everywhere, around us, even inside us.
Yeah,
We've got a stack of research and notes here, and our mission really is to unpack the key computational strategies. How do we make sense of this hidden world?
exactly. It's pretty amazing, isn't it? You can take a sample, maybe some soil, maybe a skin swab,
Right.
and with some pretty clever computational techniques, you get this detailed picture, who's there, and crucially, what they might be doing.
Okay, fantastic. So let's make sure everyone's on the same page right from the get-go.
Yeah.
Can you maybe break down what we mean by bioinformatics
Sure.
first, then maybe narrow it down to microbial bioinformatics, and finally explain that buzzword we hear so much,
Oh, yeah.
metagenomics?
Okay, certainly. So bioinformatics at its heart is really about using computers, computational tools, to make sense of biological data. It could
Yeah?
be anything, really. Your gene sequences, protein structures.
Gotcha.
Now, when we zoom in specifically on microorganisms, bacteria, fungi, viruses, all those tiny things, that's microbial bioinformatics. We're using that computing power for information about them.
Makes sense.
And metagenomics, well, it takes a slightly different angle. Instead of focusing on just one type of microbe, it's about looking at the entire collection of genetic material, all the DNA,
Oh,
from all the microbes in a single sample. Think of it like walking into a really crowded room and somehow being able to read little snippets from everyone's instruction manual all at
I
once.
like that.
That's kind of what metagenomics lets us do with these microbial communities. And the key thing is we don't need to isolate and grow each one in a lab dish.
That's a fantastic analogy. So metagenomics gives us this broad snapshot of the whole ecosystem.
Precisely.
Now, say we've got our sample, let's say, from the gut maybe. What's the very first step in turning that physical sample into useful digital information?
Right. So the journey starts with collecting the sample, obviously, and then extracting the DNA from all the microbes living in it. And this extraction step, it's absolutely critical.
What's that?
Well, you need high-quality DNA.
Yeah.
It needs to be as intact as possible and, crucially, free from contamination. You don't want DNA from the host or from the environment messing things up.
Like needing clean ingredients before you start cooking.
Exactly like that. Good analogy. Once we have that good quality DNA, the next really big step is reading its sequence. And that's where next generation sequencing, or NGS, technologies come in.
Right. NGS, we hear about different types all the time. What are some of the main ones used in microbiome studies? And does it, matter which one we choose?
Yeah, great question. So technologies like Illumina are very popular. They generate just massive amounts of DNA sequence data, and it's very accurate. The catches, the sequences, the reads tend to be relatively short, like short sentences.
Okay, short but accurate.
Exactly. Then you have other platforms like PacBio and Oxford Nanopore. These guys can produce much, much longer reads, sometimes like whole paragraphs or even pages of genetic code in one go.
Wow.
Initially, these longer reads maybe had slightly higher error rates, but the tech is improving really fast.
So why choose one over the other?
It really depends on your research question. Illumina is great for getting a good overview, like a census of who's there. But those long reads from PacBio or Nanopore, they're incredibly valuable if you want to piece together complete or near complete genomes of individual microbes from that complex mix.
Ah, like assembling a puzzle.
Yeah.
Longer pieces make it easier.
That's a perfect way to put it. It helps see the bigger picture more easily.
Okay, so we've used NGS, and now we've got this massive flood of raw DNA sequences, these short or long snippets. It sounds like we can't just jump straight into analyzing them, though. What's the next essential step?
You're absolutely right. That raw data, it needs to go through a really rigorous cleaning process. We call it quality control, or QC. It's a vital step. You need to remove any unwanted sequences that could mess up your results later on, kind of like proofreading a document for typos before you start analyzing its meaning.
Okay, so what kind of unwanted stuff are we talking about removing?
Well, we're looking for things like low-quality reads. These are sequences where the machine wasn't very confident about the letters, so there's a higher chance of errors, like blurry words in that document.
Got it.
We also need to get rid of adapter sequences. These are little bits of artificial DNA that get added during the sequencing lab process. Think of them as like technical tags or scaffolding that we don't need anymore.
Okay.
And finally, we want to remove any other contaminants DNA that might have snuck in during sample collection or the DNA extraction itself.
So basically tidying everything up so we're only left with the real microbial DNA. Are there specific software tools that bioinformaticians use for this cleanup?
Oh, yes, definitely. There are several essential tools. FastQC is one that's very widely used. It doesn't do the cleaning itself, but it takes a first look at the overall quality of your raw sequencing data.
So it tells you if there's a problem.
Exactly. It generates reports, nice visualizations that help you spot potential issues. Then, tools like Trimmomatic actually step in to do the cleaning.
Oh,
Trimmomatic can automatically find and remove those adapter sequences. It can trim away the low-quality ends of the reads, like cutting off the smudged edges of our document pages.
right.
And it can even discard entire reads if they're too short or just consistently low quality. It basically ensures we're working with the cleanest, most reliable data possible for all the analysis that comes next.
Right, that makes perfect sense. Garbage in, garbage out, as they say.
Precisely.
So, okay, we now have our pristine set of high-quality microbial DNA sequences. What's the next big question we typically want to answer about our microbiome sample?
Well, the first major question is usually, who's actually in there? Who are the players in this microbial community?
Right.
In other words, what different types of microbes are present in our sample? And this is where taxonomic classification comes in, identifying the microbes.
So how do we actually go about identifying these microscopic inhabitants just from bits of their DNA?
The fundamental idea is comparison. We compare our cleaned DNA sequences against these massive digital libraries called reference databases.
Okay.
These databases contain the known DNA blueprints, complete or partial genomes of thousands upon thousands of microbes, all carefully curated by scientists worldwide.
So you're looking for a match.
Essentially, yes. If we find sequences in our data that are very similar or identical to sequences in these databases, we can infer the identity of the microbes in our sample. It's a bit like matching fingerprints found at a scene to a database of known fingerprints.
Okay, that makes sense. And are there specific tools that are particularly good at this kind of microbial ID?
Absolutely. There are several workhorses. QIME2, that stands for Quantitative Insights into Microbial Ecology 2.
Quite a mouthful.
It is, but it's a very popular and really comprehensive software platform. It does lots of microbiome analysis, and it has very robust methods for this taxonomic classification step.
Okay, QIIME2. What else?
Then there's Metaflan, that stands for Metagenomic Phylogenetic Analysis. It takes a slightly different approach. It looks for a specific set of unique marker genes.
Marker genes.
Yeah, think of them as specific DNA sequences that are really good identifiers for particular species or groups. Metaflan uses these markers to figure out the microbial makeup, often down to the species level.
Interesting. Any others?
And then there's Kraken. Kraken is really known for its speed. It's great for assigning taxonomic labels to reads in very large metagenomic data sets. It does this by comparing short sequences, called primers, from the reads to a huge database built from complete genomes.
The speed sounds useful with all that data.
Definitely. So each of these tools, QIME2, Metaflan, Kraken, they have different algorithms, different strengths, but they're all aiming to answer that crucial first question: who is there?
Okay, so now we have a list, a sort of roster of all the different microbes in our sample, the who. But like you said earlier, that's only part of the story, right?
Yeah.
We also really want to understand what these microbes are actually doing.
Precisely. That's the next critical layer. Moving beyond just identifying the inhabitants, we want to get at the functional potential. What are they capable of doing? This is called functional analysis.
Okay.
We want to know, what kinds of genes do they possess? What biochemical pathways might be active? What roles could they be playing in that environment?
So how do we figure that out from the same DNA sequences? Does the DNA tell us about function too?
It does, indirectly. We analyze the sequences from the metagenome to predict the presence of genes that code for specific functions. It involves, again, comparing our sequences to databases, but this time, databases of known genes and protein families with known functions.
It's different databases.
Exactly. So if we find sequences in our metagenome that look very similar to genes known to be involved in, say, breaking down a specific type of sugar or maybe resisting an antibiotic,
Right.
we can infer that our microbial community likely has the potential to perform those functions. It's a bit like looking at a recipe book, our metagenome, and figuring out what kinds of dishes could potentially be made based on the list of ingredients, the genes, that we find.
That's a neat way to think about it. So what are some of the key tools for this kind of functional prediction? Are there equivalents to CRY-ME2 or Kraken for function?
Yes, there are. Two prominent tools often used are Human N2 and Megan.
Human N2.
Yeah, like human.
Sort of. It stands for HMP, Unified Metabolic Analysis Network 2. It's specifically designed to figure out the functional profiles of microbial communities, often down to the species-level contribution. It tries to reconstruct which metabolic pathways are present and estimates how abundant they are. Okay, so very focused on metabolism. What about Megan?
Megan, that's a metagenomic analyzer, takes a bit of a broader approach. It's quite versatile. It can do both taxonomic and functional analysis. It works by mapping the sequence reads onto nodes in a big hierarchical tree. This could be the tree of life for taxonomy or a tree of biological functions.
So it gives you both who's there and what they might be doing.
Yes, it provides a really comprehensive overview of the community's capabilities alongside its composition. Using tools like these, for instance, researchers have been able to see how the gut microbes of people on different diets have different sets of genes for breaking down specific carbohydrates.
Ah, connecting diet to microbial function.
Exactly. It gives us real insights into that complex interplay.
This is fascinating. It's like we're piecing together this incredibly complex puzzle. We start with millions of tiny DNA fragments,
Right.
and we figure out who they belong to and now what functions they might have. You mentioned earlier about piecing reads together, like into longer sentences or paragraphs. Assembly, you called it.
Yes, that's assembly. It's another really important step, especially if you want deeper insights. When we have enough overlap between our sequencing reads, particularly useful with the shorter reads like from Illumina,
Okay.
we can use computational algorithms to stitch them together, like solving a jigsaw puzzle.
Yeah.
This creates longer, continuous sequences of DNA, which we call contigs.
Contigs. Got it.
And in some cases, especially if we have good coverage or if we started with those longer reads from PacBio or Nanopore, we can assemble these contigs even further, sometimes getting near complete or even complete microbial genomes out of the mix.
From the whole community sample.
Yes, these are called metagenome assembled genomes, or MAGs for short.
MAGs. Okay. That sounds incredibly complex to pull off.
Yeah.
What's the big advantage of going through all that effort of assembly? Why not just stick with the reads?
Well, assembling longer stretches of DNA, and especially getting those MAGs, allows for much more in-depth genomic analysis of individual community members.
How so?
With a MAG, you have a much better picture of all the genes present within that specific microbe. You can reconstruct its potential metabolic pathways more accurately, figure out its evolutionary relationships, maybe identify genes for virulence or antibiotic resistance that are specific to that organism.
Also, you get a more complete instruction manual for individual microbes.
Exactly. It's like being able to read an entire chapter or even the whole book for a few key players instead of just scattered sentences from everyone. It gives you a much richer understanding. For example, resolving tricky, repetitive regions in bacterial genomes, which is hard with short reads, is much easier with MAGs, and that's been key to understanding genes involved in causing disease.
Makes sense. So what are the workhorse tools for assembly? The software that puts these fragments back together.
Several powerful assembly tools are out there. MegaHit is a really popular one, especially for very large and complex metagenomic data sets. It's known for being fast and efficient, particularly with short reads.
Mega hit, okay.
Then there's SPADES, S-P-A-D-E-S, that stands for St. Petersburg Genome Assembler. It's another widely used assembler, very versatile. It can handle data from single cells, isolates, and metagenomes, and it often produces really high-quality assemblies.
Spades, got it. And okay, let's say we've run Spades or MegaHit and we have a whole bunch of these longer contigs. How do we actually figure out which contigs likely belong to the same microbe to build those MAGs you mentioned? How do we sort them?
Ah, that's the next challenge. That sorting process is called binning. Binning is the computational process of grouping the assembled contigs into clusters, where each cluster hopefully represents the genome of a single microbial species or strain.
Binning. So how does the computer know which contigs belong together?
It usually relies on various characteristics of the contigs themselves. One key thing is their DNA composition. For example, the percentage of guanine and cytosine bases the G's and C's in the DNA code, often called GC content. This percentage tends to be relatively consistent within a single species, but varies between different species.
Like a genomic signature?
Sort of, yeah. Another important clue is coverage. That means how many sequencing reads mapped back to that contig. Contigs from the same organism should generally have similar coverage levels across different samples, assuming the organism's abundance doesn't change drastically.
Okay, so composition and coverage patterns.
Exactly. Algorithms use these features, sometimes along with other information like the frequency of short DNA words, like sequences of 4 bases called tetranucleotides, to group contigs together.
It still sounds like a tough sorting job. What are the go-to tools for binning?
There are several really effective binning tools available now. MetaBat is one. It stands for metagenome binning using abundance and tetranucleotide frequencies. As the name suggests, it uses both how abundant contigs are across samples and those short tetranucleotide frequencies to do the grouping.
Metabat. Okay.
Another popular one is CONN-CICT. That stands for clustering contigs using coverage and composition. As its name implies, it primarily relies on how the coverage of each contig varies across multiple samples, along with DNA composition to perform the binning.
Con and COCT.
Right. These tools are really crucial for helping us reconstruct individual genomes from that incredibly complex mixture of DNA fragments we started with.
We've covered so much ground already. From the raw sequence reads, cleaning them up, figuring out who's there, what they might do, even assembling their genomes.
It's quite a workflow.
It really is. But often, the goal isn't just to describe one sample, right? We want to compare microbial communities, maybe between healthy people and sick people, or different soil types, or before and after a treatment. How do we do that comparison in a meaningful way using bioinformatics?
That's a huge part of microbiome research, absolutely. This is where statistical analysis and the concept of ecological diversity really come into play.
Okay.
We use a whole suite of statistical methods to compare these communities across different samples or conditions. And the programming language R, along with its incredibly rich ecosystem of specialized packages, is a real workhorse in this area for bioinformaticians and ecologists.
All right, I've heard of that. Are there specific R packages for microbiome data?
Oh yes. Packages like phylasec and vegan are extremely popular. They provide a massive toolbox for importing microbiome data, calculating various diversity metrics, visualizing similarities and differences between communities that's often done using a technique called ordination, and performing statistical tests to see if the differences we observe between groups are actually statistically significant or if they could have just happened by chance.
Okay, phylasec and vegan and R. You mentioned diversity. When we talk about diversity in microbiomes, what exactly are we measuring? Is it just the number of different types?
That's part of it, but it's more nuanced. We generally look at two main facets of diversity: alpha diversity and beta diversity.
Alpha and beta. Okay, what was alpha?
Alpha diversity refers to the diversity within a single sample. It's a measure of the complexity inside one specific community. It considers two main things: richness and evenness.
Richness and evenness.
Right.
Richness is simply the number of different types of microbes present. How many distinct species did we find in that sample? Evenness describes how similar the abundances of those different types are. Is the community dominated by just one or two types, or are many different types present in roughly equal numbers?
Ah, okay, like a garden with many different types of flowers,
Right.
richness. And are they all planted in similar numbers, or is it mostly roses, evenness?
But that's a great analogy, exactly. And there are different mathematical ways to measure alpha diversity. SOBS, for instance, is just the raw number of species, the observed richness. Chaff1 tries to estimate the total richness, including species we might have missed because they're rare.
Okay.
Then you have indices like the Shannon Index and the Simpson Index. These incorporate both richness and evenness into a single number. Shannon gives more weight to richness, while Simpson is more influenced by the most abundant species.
So different ways to quantify that within sample diversity. Precisely. And there's also phylogenetic diversity, or PD. This one actually considers the evolutionary relationships between the microbes. A sample with microbes spread out across the tree of life might be considered more diverse, perhaps more resilient, than one where all the microbes are very closely related.
Interesting. Okay, that's alpha diversity within a sample. What about beta diversity?
Beta diversity measures the diversity between samples. It quantifies how different the microbial community composition is from one sample to another.
So how similar or dissimilar are two communities?
Exactly. It's often expressed as a measure of ecological distance or dissimilarity between pairs of samples. Common beta diversity metrics include Bray-Curtis dissimilarity, which looks at the relative abundance of each species in two samples.
Okay.
There's also the Jacquard index, or Jacquard distance, which only cares about presence or absence. Does a species exist in both samples, just one or neither? It ignores abundance. And then there's a really important family of metrics called unifrac distance. Unifrac is powerful because it incorporates the phylogenetic relationships between the microbes, just like phylogenetic diversity did for alpha diversity.
So it considers how related the microbes are when comparing communities.
Yes. And unifrac comes in two main flavors: unweighted and weighted. Unweighted unifrac focuses just on which lineages are present or absent, so it's sensitive to changes in rare members of the community. Weighted unifrac also takes the abundance of each lineage into account, so it reflects shifts in the dominant members of the community.
Wow, lots of options there too.
There are, and it's often really informative to calculate and look at several different alpha and beta diversity measures because they capture different aspects of the community structure and composition. You get a more well-rounded picture that way.
That makes sense. Use multiple metrics for a fuller understanding. Now, I've also heard people talk about something called rarefaction when discussing diversity. What's that all about? Is it another metric?
Rarefaction. It's not exactly a metric itself, but it's a crucial statistical technique used often before calculating diversity metrics, especially alpha diversity.
Okay, why do we need it?
The issue is that the number of species you detect is often dependent on how deeply you sequence the sample. If you sequence one sample much more than another, you're naturally likely to find more rare species in the deeper sequenced one, just by chance.
Right, more sampling effort finds more rare things.
Exactly. So if you directly compare, say, the observed richness SOBs between these two samples, the deeper one will almost always look richer, even if the underlying communities have similar richness. It's an artifact of sequencing depth.
So rarefaction fixes that.
It attempts to level the playing field. Rarefaction involves randomly subsampling reads from each sample down to the same, usually lower, number of reads, typically the depth of the shallowest sample. You then calculate your diversity metric on this subsampled or rarefied data.
So you compare apples to apples in terms of sequencing effort.
Precisely. It allows for a fairer comparison of diversity, particularly for metrics like richness that are very sensitive to sample size. We often plot rarefaction curves where we repeatedly subsample at different depths and plot the diversity to see if we've sequenced deeply enough to capture most of the diversity.
Okay, that's a really important consideration for fair comparisons. So we've done all this analysis, taxonomy, function, diversity comparisons. We have numbers, distances.
Yeah.
How do we actually see this information? Visualization must be key.
Absolutely essential. Visualization is how we make sense of these incredibly complex, high-dimensional data sets. Staring at tables of numbers only gets you so far.
So what kinds of plots do people use?
There's a whole range. Heat maps are really common and powerful. They can show the relative abundance of, say, different bacterial genera across dozens or hundreds of samples. You use color intensity to represent abundance.
Right, like a grid with different colors.
Exactly. Stacked bar plots are another classic. Each bar represents a sample, and the bar is divided into colored segments, where the size of each segment shows the proportion of a different microbial group, like phylum or genus. It gives you a quick overview of the major components.
Okay, good for composition. What about showing how samples relate to each other based on beta diversity?
That's where ordination plots come in. Techniques like principal coordinates analysis or PCOA take those complex beta diversity distance matrices we calculated.
Right, the Bray-Curtis or unifrac distances.
Exactly. And they try to represent those distances in a lower dimensional space, usually 2D or 3D, that we can actually plot. Samples that are close together in the PCOA plot have similar microbial communities, according to the distance metric used, while samples far apart are dissimilar.
So you can visually see clusters of similar samples.
Precisely. It's a great way to explore overall patterns and see if samples group according to factors you're interested in, like treatment group or environment type.
Are there tools that make exploring these visualizations easier, maybe interactive ones?
Yes, definitely. Static plots are great for papers, but interactive tools are fantastic for exploration. Emperor is a popular tool specifically designed for visualizing and interacting with ordination plots like PCOA.
Emperor, like the ruler.
Exactly. It lets you rotate the 3D plots, zoom in, color the sample points based on different metadata like diet or health status, identify outliers, and really get a feel for the structure in your data.
Cool. Any others?
Another interesting one is Caler. Caler focuses more on interactive heat map exploration. It links the heat map directly to the sample metadata and phylogenetic tree, making it easier to spot which specific microbes might be driving the patterns you see, or how groups of related microbes behave across samples.
So these tools help you dig into the patterns interactively.
They do. They're invaluable for generating hypotheses that you can then test more formally with statistics.
We've covered such a huge amount, from the basic DNA sequence all the way to these sophisticated visualizations and interactive tools. But I know this field is always moving.
Yeah.
What are some of the more advanced analyses or future directions people are working on in microbiome bioinformatics?
Yeah, it's a very dynamic area, constantly evolving. One area is the refinement of statistical methods for what's called differential abundance analysis.
Differential
Yes.
abundance.
Basically going beyond just saying communities are different, beta diversity, and trying to pinpoint which specific microbes or functional pathways are significantly more or less abundant between different groups of samples, like cases versus controls.
Finding the key differences.
Exactly. And this is trickier than it sounds because microbiome data is compositional. The abundances are all relative proportions that sum to one. So if one thing goes up, something else has to go down.
Hmm.
Traditional stats methods aren't always ideal for this. So specialized tools are being developed to handle this compositional nature properly. Linda is one example of a more recent method designed for robust differential abundance testing in microbiome data.
Okay. Better stats for compositional data. What else is cutting edge? I hear a lot about machine learning everywhere. What's it big in microbiomes too?
Oh, absolutely. Machine learning is becoming increasingly important. It's being used to find complex patterns in the data that might be hard to spot otherwise, and importantly, to build predictive models.
Predictive models, like predicting disease.
Potentially, yes. Techniques like random forests, which are a powerful type of machine learning algorithm, are used quite often. You can feed these models information like the taxonomic profiles, functional pathway abundances, maybe even phylogenetic information.
All the stuff we've been talking about.
Exactly. And train the model to predict an outcome. That could be classifying samples into disease states, predicting response to a particular drug or diet, things like that.
That sounds really powerful. Are there challenges?
Definitely. You need to be careful about how you prepare the data, how you select the most informative features for the model, and critically, you need to rigorously evaluate how well the model performs. We use techniques like cross-validation, often nested cross-validation for unbiased assessment, to estimate how well the model will generalize to new, unseen data.
Okay.
And we often visualize the prediction performance using things like ROC curves, which help us understand the trade-offs between correctly identifying true positives and avoiding false positives. Researchers have already used these approaches, for instance, using gut microbiome data and random forests, to build models that can predict the onset of certain inflammatory diseases with pretty good accuracy.
Wow. So moving towards prediction and maybe even diagnostics based on the
But
microbiome.
it's certainly a major goal for many researchers, yes.
And are people combining metagenomics with other types of omics data for an even deeper view?
Yes, absolutely. That's another major trend, multi-omics integration. To get a truly holistic picture, we often need more than just the DNA blueprint from metagenomics.
What other omics are relevant?
Well, there's metatranscriptomics. That looks at the RNA molecules being transcribed from the DNA. It tells us which genes are actually active and being expressed by the microbes at that particular moment, rather than just which genes are present.
So function in action.
Kind of, yes. Then there's metaproteomics, which looks directly at the proteins being produced, the actual molecular machinery doing the work in the cells.
Okay.
Single-cell genomics is also advancing rapidly. This lets us sequence the genome of individual microbial cells from a complex sample, which can reveal variation within populations that gets averaged out in bulk metagenomics.
Wow. Down to the single cell.
And finally, metabolomics. This focuses on the small molecules, the metabolites, that are the inputs and outputs of microbial metabolism, things like sugars, amino acids, short-chain fatty acids. This gives a snapshot of the overall biochemical activity of the community and its interactions with the host or environment.
So combining DNA, RNA, proteins, metabolites.
Yeah.
That sounds like a complete systems view.
That's the goal. Each omic layer provides unique insights, and integrating them gives a much richer, more comprehensive understanding. For example, combining metagenomics and metatranscriptomics helps us link genetic potential to actual gene activity. It's complex, computationally, but really powerful.
And you mentioned tools for study design too.
Yes, thinking about statistics early is crucial. There are tools like mPower, which helps researchers estimate how many samples they'll likely need for their study. That's called power calculation, to have a good chance of detecting statistically significant differences in the microbiome, given the expected variability.
Planning ahead statistically. Smart. So looking forward, what are the big frontiers?
I think we'll continue to see development of more accurate, more scalable algorithms to handle ever-growing data sets. Databases will become larger, better curated, and more comprehensive. And a big push is towards developing integrated platforms and methods that can seamlessly analyze multiple omics data types together.
Making multi-omics easier.
Exactly. Plus, ongoing improvements in sequencing technology itself, like long-read methods becoming both longer and more accurate, will keep opening up new possibilities, like assembling genomes of viruses within metagenomes or resolving really complex structural variations in bacterial genomes. It's a really exciting time to be in the field.
It certainly sounds like it. This has been an absolutely incredible deep dive. Before we wrap things up, maybe you could just give us a quick rundown of some of those key bioinformatics terms we've touched upon today, just to help solidify them for us and the listeners.
Certainly. Happy to.
Great. Let's start with gene.
Okay, a gene. It's basically a specific segment of DNA that holds the instructions for building something functional, usually a protein or maybe an RNA molecule. Think of it as a single recipe in the big cookbook of the genome.
Okay. And a genome.
The genome is the entire set of genetic material, all the DNA in an organism. It's the complete instruction manual, the whole cookbook.
Got it. Metagenome.
The metagenome, as we discussed, is the collective genomic material from all the different organisms found in a particular environmental sample. So it's like having all the instruction manuals from all the different microbes living together all mixed up.
Right. Accession number, like from GenBank.
An accession number is just a unique ID code. It's assigned to a specific sequence record when it's submitted to a public database like GenBank. It's like the unique library call number or ISBN for that particular piece of genetic information.
Make sense? Alignment.
Alignment, or sequence alignment, is the process of lining up two or more sequences, DNA or protein, to highlight regions where they are similar. These similarities can suggest that the sequences share evolutionary history or have similar functions or structures. It's like comparing sentences to see which words match up.
Okay. Annotation.
Annotation, specifically genome annotation, is the process of identifying important features within a DNA sequence. This means finding where the genes are, what they might do, locating regulatory elements, etc. It's like adding labels and explanations to a map to show what all the symbols mean.
Great. Contig.
A contig is a continuous stretch of DNA sequence that's been assembled by computationally overlapping shorter sequencing reads. It's a longer piece built from smaller fragments, like putting sentence fragments together to make a longer clause.
Domain in the protein context.
A protein domain is a distinct region within a protein structure that often has a specific function and can sometimes fold and work independently of the rest of the protein. Think of it like a specialized module or tool within a larger machine.
Okay.
E-value.
The E-value, or expectation value, is a statistic you get when searching sequence databases. It tells you the number of hits you'd expect to find by random chance that are as good as or better than the match you actually found. So a very small E-value, close to 0, means your match is highly unlikely to be random chance. It's probably biologically significant. Right, lower is better. EST.
And EST stands for expressed sequence tag. It's a short piece of DNA sequence that's been copied from a messenger RNA, mRNA, molecule. Since mRNA is the template for making proteins, ESTs give us a snapshot of which genes were actively being expressed or turned on in the cell or tissue where the mRNA was collected.
Okay. Homolog.
A homolog is a gene that's related to another gene because they both evolved from a common ancestral gene. Homologous genes, or proteins they code for, often have similar sequences and may perform similar functions, sometimes in different species, like related words in different languages, for example, night and nacht.
Got it. And finally, motif.
A motif, usually referring to a protein motif, is a short, conserved pattern of amino acids within a protein sequence. These patterns are often associated with a particular function, like binding to DNA, or a specific structural feature. It's like a recurring signature or pattern found in different proteins that hints at their job or shape.
That's fantastic. A wonderfully clear tour through some key bioinformatics vocabulary. Thank you so much for joining us and sharing all this incredible detail on bioinformatics.
It was absolutely my pleasure. It's a field I'm really passionate about.
We've really explored the computational strategies, the tools, the challenges, everything that's revolutionizing how we understand this microbial world. We really hope this deep dive has given you, our listeners, some valuable insights. Insights into how this vast amount of sequencing data gets transformed into real knowledge about these microbial communities, their functions, and their impact on, well, everything from our health to the planet's health.
It's truly a fundamental part of biology now.
And remember, this field is moving so fast. New tools, new algorithms, new approaches are emerging constantly. We definitely encourage you to maybe look up some of the tools or concepts we mentioned, explore the research, and just appreciate the amazing possibilities that microbiome bioinformatics holds for future discoveries.
Absolutely. The potential is huge, and there's always more to learn.